Okay, so in section 4, we're going to talk about chemical controls of microbial growth. So before we talked about physical methods, and those are important. But when we're talking about everyday disinfection, we're really talking about chemical agents. And these agents are going to target different things like cell walls, plasma membranes, proteins, and nucleic acid. But their performance is going to depend on their environment. So it's going to depend on what temperature they're in, the pH, the contact time, so how long we need to apply the chemical in order for it to be effective, the concentration of the chemical, organic load, such as like blood or feces, and the freshness of the solution itself. So we've taken these different chemicals and they're kind of grouped into 9 major categories. So that's what we're going to go over now. So our first category is going to be phenol or phenolics. And this kind of began with Joseph Lister. If you remember him from lecture exam 1, he introduced phenol, which is also known as carbolic acid, back in the 1860s, 1870s, somewhere around there. And he introduced it to help reduce surgical infections. And we still use phenolic compounds today. We have just chemically modified them to help boost their antimicrobial power. We've also modified them so they're not as stinky or as odorous, and they're not as irritating. And how they work is they denature proteins and disrupt membranes, and they even work in the presence of organic material like blood or feces. So this makes them incredibly useful in healthcare and laboratory settings. One example of a phenolic is, bisphenolic is triclosan. So triclosan used to be in a majority of soaps and household products, but back in 2016, the U.S. Food and Drug Administration actually banned its use in over-the-counter products like hand soaps, because there wasn't enough evidence of its added benefit, and that's when concerns of resistance started to become known. So this is important. We'll talk about how things are removed or regulated in order to kind of control resistance to certain pathogens. We'll talk about a little bit at the end of this section and a lot in the next chapter. Another category is alcohol, so things like ethanol and isopropanol. These are really fast acting. They're what we consider intermediate level disinfectants, and they work by denaturing proteins and disorganizing membranes. Their optimal concentration is 70 to 90%. So usually you can't go to Walgreens and buy isopropyl alcohol and 100% concentration. Why? Because pure alcohol works less effectively than the 70-90% concentration because water is required for protein denaturization. If you remember back when we had to break down our macromolecules, we talked about hydrolysis is how we break them down. Water is required. So that's why normally it's either 70 to 90% alcohol and then 10 to 30% water. But alcohols are important because they can kill vegetative bacterial cells, fungi, envelope viruses, but they don't reliably kill inactive spores or even some of the non-envelope viruses. Some good things about them is they evaporate very quickly and they don't leave any residue. So that is beneficial when you're talking about clinical use for them. Okay, another category or group is going to be our halogens. So if you remember your chemistry or periodic table, your halogens are your iodine, chlorine, bromine, and fluorine. These also act at the intermediate level and they unfold and denature proteins, and they are pretty versatile. You don't need to know the specifics about how all of these act just collectively, but one of the most important ones is iodine because it's used for skin prep. Chlorine's really important when we're talking about its form in bleach or its form in chlorine dioxide, because chlorine's used a lot for wastewater disinfection or just regular water disinfection. It's also used to sanitize hospital equipment. Bromine is mostly used when we're talking about hot tubs because it volatilizes less rapidly at high temperatures. So it's stable at high temperatures. So instead of using chlorine, they use bromine. Fluoride, this is typically used when we're talking about dental plaque. Now it's a little bit controversial, but usually most toothpaste has fluoride, or you get some sort of fluoride treatment after cleaning, something like that. Okay, we also have oxidizing agents, so things like hydrogen peroxide, oxone, and peracetic acid, and these kill by oxidizing enzymes and releasing oxygen radicals, which can be harmful. Hydrogen peroxide is really helpful for sterilizing surfaces, but if we're talking about microbes and on us or in wounds, it is helpful, but it is quickly broken down because we have catalase. Ozone is really effective. It's a little bit expensive, but it's usually used as a disinfectant for water treatment, and not in the US. This is actually in Canada and Europe. They use it, but it's pretty good for water. Peracetic acid, this is what is considered a high-level sporicide. So it's really important when we're talking about food or healthcare because it works even in the presence of organic soil and it doesn't leave any toxic residue. Surfactants, we have surfactant in our lungs. If you remember from the respiratory chapter in Anatomy and Physiology 2, so surfactants' goal are to lower surface tension. And it does so by helping wash away oils and microbes. So soaps are a good example of a surfactant. They primarily de-germ by emulsifying oils rather than killing microbes directly. You also have quaternary ammonium compounds, also called quats. These disrupt membranes and they're commonly found in things like mouthwashes, some of the chemical wipes that they use in like a dental office or a hospital. They're part of a lot of hospital cleaning protocols. That's all I really want to say about that. Okay, heavy metals. Heavy metals are things like silver, mercury, copper, and zinc, and they bind to a lot of sulfurs that are found in proteins, causing them to be denatured. Copper's fairly frequently used, not only for microbial control in hospitals or clinical settings, but also for talking about agriculture. So a lot of the antibiotics or the fungicides that are applied to a lot of the trees or crops are actually applied in conjunction with copper. Another thing I think I read not that long ago, it might have been 10 years ago now I think about it, but copper is commonly used in space now. So like their toilet seats have a copper coating to help prevent microbial growth and things like that. So there's a lot of interesting things they're doing by incorporating some of these heavy metals. But again, most of these are in conjunction with other things. So it's these heavy metals in conjunction with antimicrobials, not just them by themselves. Aldehydes. So aldehydes act by cross-linking different functional groups. So things like amino groups, hydroxyl groups, sulfhydryl groups, carboxyl groups. And when they cross-link these groups, they inactivate the proteins that they're associated with or the nucleic acids that they're associated with. And there's two examples there, but I'm not super concerned with those. Okay, gaseous agents is another one. One common example of a gaseous agent is ethylene oxide. And it's used a lot for sterilizing heat sensitive or moisture sensitive items. So things like catheters, a lot of electronic equipment, components of different machines involved in hearts or lungs monitoring. And also ethylene gas is used to sterilize mattresses. So it's really important because it can kill even spores and through cross-linking and pretty much any microbe. But I will say the process takes a really long time. It can take hours. And after you apply it, you need to also allow for a period of like aeration to kind of get it out. It was very, very frequently used in hospitals and dental offices as a source of chemical control until they correlated ethylene oxide with cancer. So in 2024, the United States Environmental Protection Agency actually enforced new ethylene oxide emission standards. And the goal of this was to reduce community cancer risk from these sterilization facilities that were using ethylene oxide, whose workers were found to have a higher level of cancer incidence than those in the general population. So now there's very strict standards in ethylene oxide use, and I would expect that these are going to be used less going forward. Okay, enzymes. These are really great because they're very precise when we're talking about their action. All enzymes are very specific for a substrate. So lysozyme, we have that in our tears. There's also other enzymes in food and wine that disrupt bacterial peptidoglycan. We also have prionizyme. This system has basically revolutionized the removal of prions from medical instruments. Really important because remember, prions are resistant to autoclaving and conventional disinfectants. They're also using a lot of enzyme cleaners and endoscope reprocessing, so that's another important use of them. But basically, medical instruments